Hello. Hi, I'm Aishwarya, and a very good evening to everyone out here. I'm really glad that I see people slowly joining. In fact, I'm hoping more people join us on the session today. The whole series that we have is realistic applications in 3D bioprinting and how we have experts or we have people working on the 3D bioprinters coming on our platform to give you insights about 3D bioprinting. So, giving you a brief about our sessions, I will start with talking about just the basics of 3D bioprinting, basic techniques, basic ideas. And after that, we will progress towards having our experts talk. So we have two people on the dais for us. One is Dr. Shreya Malhotra, who is a researcher, a PhD scholar, who is working towards cardiac tissue engineering, that is cardiac 3D bioprinting, as well as Dr. Kaushik Chatterjee, who is going to help us and tell us about the whole idea of using of polymers in the field of 3D printing, 3D bioprinting, as well as his experiences using the machines. I hope to dwell better, and I hope to see more people take this opportunity to get forward. On YouTube, I would really request everybody to like, share, subscribe, and now I'll begin the session. So I'm going to share my screen. Yeah, so coming to the point, the first topic being the potential of bioprinting in life sciences and healthcare. So my name is Aishwarya again, and I'm a technical specialist at Altum Technologies. So talking about Altum Technologies, we are a 3D innovation platform company, and we have several types of solutions in our hand, that is Dassault Systems, MSC Software, Stratasys, Artex 3D, Biovia, Celic. And we are a decade or more old company, and we've been setting our foot in the industry since then. We have various partners, various solutions, a team of entrepreneurs, salesmen, scientists, and any other area required in terms of technical expertise. So coming down to our customers, we actually have a very large list. So this is just a partial list. So we have people from the education and research, we have aero and defense, automotive suppliers, healthcare, life sciences, consumer products, automotive, aerospace, etc. So we have a large list of people coming from various sectors, that is companies, education and research, defense, whatever sectors you would say, we have solutions provided to all of these sections. So coming down to the main point, I will brief you about what biomedical devices are, what biomedical engineering is, for you to understand how well a tool could connect in terms of this application. So talking about biomedical engineering, it is an interdisciplinary field which consists of engineering concepts, medical concepts, life science concepts, and many other such areas, which together aim at providing you solutions for a specific problem, and that is in the healthcare space or in the life science space. And now medical devices have quite a large set of categories, and some are assistive, some are aiding, some are invasive. So we will dwell a little bit over there and see how we come to the part or the stage where a more recent technology like 3D bioprinting can help us in various applications. So the kind of biomedical devices that we see of late, or at least what we would recognize generally, would be an MRI device, a CT device, or any hospital equipment device that you see in general, maybe a life care monitor, a healthcare monitor, a ventilator, and so many such applications or so many such machines. Even the most basic things like a stethoscope, ECG and EKG, a weighing balance, a weighing machine, all of these type of equipments come under biomedical devices, which are ideally meant to aid any sort of healthcare requirement. This is another one, which is the use of biomedical devices more on an invasive level, that is For example, you have defibrillators, you have gastric simulators, insulin pumps, cochlear implants, knee implants, eye lenses, stents, devices, silicone implants, screws and gauges. And these are the type of devices that we would have on a more invasive note, something which is rather more than an assistive. It is something that will be a replacement or a regenerative structure that can add value or add to your existing deformity. The next one is use of AR, VR, imaging techniques, imaging tools, which is again another sector. So you have several areas where we dwell into. So I will be talking about the fourth one, which you see here, that is the cochlear implants for ears, where a specific tool would work. So any of these devices, while being created, requires a set of manufacturing standards or a manufacturing method, right? Either they would be a specific um, basic method like molding or etching or anything of that sort, where you try to create specific design from a main master copy and then you try to replicate it. But there are also cases where you can create easily disposable master copies, and also you can create customizable devices. So customizable devices is where 3D printing comes into picture. So talking about what 3D printing is. So it is a form of additive manufacturing, and this is a process where you're making three-dimensional physical objects from a digital file. So you're taking something called an STL file, an OBJ file, and you're using these files and eventually trying to create the same object which was already on a soft file. Basically, a soft copy would be converted into a hard copy, is what you would say, is where you have a three-dimensional structure that is created. So here you will see that whatever additive manufacturing technique works by, it requires layer by layer printing. So you have the material being laying down on a successive manner in such a way that it creates that structure that you want. And this is a tool with which can efficiently allow you to produce complex shapes and sizes. And the concept of this is more widely seen in the mechanical sectors, where you're printing with plastic materials, polymer materials, metals, and you can incorporate these concepts into later parts of your work or the techniques or technology that we're talking about. Now, when you talk about 3D printing, we would have two types of areas that we will ponder into. One being generally 3D print in the healthcare space or the life science space, where you have maybe invasive devices, form and fit testing devices, wearable devices, patient-specific invasive devices, or surgical tools or guides, and then you have general devices or design prototyping. So this comes under a sector where you have something called 3D scanning as well as 3D printing, which can efficiently help in the healthcare section. And you have also the bioprinter. And this is something which helps in various areas of research or various steps that we take in terms of technological advancements towards disease management or trying to improve a specific situation. So you start with software robotics, biomaterial development, tissue engineering, organ on a chip, 3D cell culture, and many more to go. So these are all images of just general examples of how general plastic 3D printing or any general device could work. So you would have metal implants, metal grafts, pre-surgical models, dental implants. Then you have prosthetic devices. So you have many sectors that can be catered to when you're using 3D printing as a technique, both bioprinting as well as general 3D printing. So talking about biomaterials, because again, when you're trying to create something, you're basically trying to create a 3D design, a hard copy of what I said of any object, and you need to know that you're using a material which is compliant, or also you're using a material which is giving you efficiency, strength, general properties that could help in making that design, making that form, and allowing it to give you the right fit of object that you want. So here the materials are basically that which can easily interact with living systems, like they do not cause any sort of negative influence on your living systems, like clots, hemorrhages, thrombosis. These are some specific terms we use when it comes to biomedical devices, because they are sometimes intact, that is over a body, non-invasive or invasive. So we have words like bioinks, that is cell-laden biomaterials specific for additive manufacturing, and biocompatibility being a topic, that is whether there is a host response in a specific application. So we are trying to maintain that. So taking a material, having the cells along with, just seeing whether there is a complementary action between the object and the material that you're using. And other materials that are generally used in healthcare or any space would be materials like PEEK or PP or general ABS materials, or you will have something called alginate, which I'll talk about. And we have also metals. So you have various types of materials that are considered when it comes to printing with various type of materials to create a specific design that helps you with your work. Now, the journey towards tissue engineering, or even the idea behind bioprinting, falls from how I mentioned using 3D printed parts or using prosthetic parts, which were bionic human structures, and we would try to create implants or prosthetic devices which would assist in terms of a human body. But now we are also trying to create a more biologically similar material or a biologically similar substitute. That is maybe sometimes examples of urethra being created. So here they have used a biocompatible material. They have used a material that will not degrade yet also give you the right properties while being in your body, not allowing any deformities or any thrombosis happening. Then you have cornea and you have bladders, you have the trachea. So you have many examples where 3D bioprinting has been incorporated or tissue engineering concepts have incorporated. So in the next session, I will be talking a little more about tissue engineering or the details about it. For now, we will dwell into what bioprinting is. And coming down in general, why do you find a specific solution is when you have a specific challenge or a problem. And the problems that we would talk about would be, say, the lack of organ transplants. That is a very big picture concept, but this is the end goal of using any type of tool, any type of technique, or even the idea of tissue engineering, because we are eventually trying to create biosimilar or bioartificial structures for the body that can effectively eradicate comorbidities or deaths caused by failures of the body, failures of the organ, a bone issue, an accident. We have many examples to give. And on the other hand, when it comes to research, we have waste due to wrong testing methods, and then we have the lab to shelf time. So whatever research techniques that are being incorporated will always be limited to one particular type of sample model or a testing model, and sometimes that process would take a lot of time. So keeping all of these gaps in mind and having this idea that, okay, we have these solutions in the healthcare and life sciences space, is where you have a tool that is a 3D bioprinter. Now, what is 3D bioprinting? Is that is a form of additive manufacturing. This uses cells and other biocompatible materials as inks to print living structures. So you are trying to create something in a layer by layer fashion in order to mimic the behavior of natural living systems. So 3D printing, tissue engineering I mentioned very lightly, and 3D printing I've also mentioned about how it is how it uses a specific idea. So here, when it comes to 3D bioprinting, you are actually using living cells. You're using bioinks, you're using culture media. Cells in the form, you would use cells in the form of spheroids or organoids or in clusters, which will give you the functioning of live cells in a cluster that will give you the right biochemical properties of the synthesis. Then you have a bioink, which is basically the foundation of what you're going to print. It's going to be the structure bearing or the physical environmental condition creating point, which is called as a bioink. And most of the work that has been going on in the bioprinting realm has always started from the bioinks, coming down to development of bioink materials, checking the interaction of a bioink with a cell, creating something which is highly mechanical, working around or playing around with the degradation rates, working around with ideas of how much mechanical strength a specific material can provide. or many such ideas in terms of rheology, viscosity, printing ability. All that is checked here. And then you have the culture media. Culture media is basically the environmental factors, say uh, the cell culture media, or you would use something like a saline medium, or you would be printing it in air, you would be printing it in a culture media itself, or you have a specific environment that you are trying to test and print your structures in. This is a small video which will give you a little more depth into what 3D bioprinting is.
3D Bioprinting by Abir Sinyal with the Newen Lab at UNC Chapel Hill. We print things all the time. Whether they're documents, posters, or flyers, printing is an important technology in our daily lives. Relatively recently, however, printing took a big step forward with 3D printing. We could build a 3D model on the computer, send it to the printer, and we could have that object in our hands in hardly any time. What if I told you that this isn't the limit? There is more to explore. What if we could print living things made up of cells? We could make blood vessels, tissues, and organs. We could help the hundreds of thousands of people waiting on transplant lists. This idea is not too far from reality if we use 3D bioprinting. 3D bioprinting is just like normal 3D printing, except it uses living cells as ink to create living structures. Like 3D printing, you have to create a model on the computer, then layer by layer, your living structure will be printed. The first step in the process is known as pre-bioprinting. Here, the desired product is determined and studied. Oftentimes, 3D scans, such as CT scans or MRI images, are taken of the desired product and are then converted into a series of 2D images to serve as a template for the different layers. Cells are then isolated and extracted from an organism, which is often the recipient of the product, in order to create bioink. Bioink is one of the most important parts of the 3D bioprinting process. It starts with the cells that were extracted. Then, molecules called hydrogels are added to provide water for the cells. Other nutrients and chemicals are added so that the cells can grow and communicate as if they were in a living body. Once the bioink is developed, we reach the step of doing the actual printing. Using the bioink, our organ is printed layer by layer until we achieve our final product. There's not just one method of bioprinting. Different types include inkjet printing, acoustic printing, and laser printing. The most common type of bioprinting, however, is extrusion printing. This method is probably how you would normally think of 3D printing. The bioink is loaded into a printing chamber and is pushed out a round nozzle. The nozzle produces a tiny filament that is often around 400 microns in diameter, or about the thickness of 4 pieces of paper. Once you've finished the physical bioprinting portion, the final step is solidification. In general, bioink is a viscous liquid, so in order for it to become the desired product, it needs to harden. Sometimes the bioink will solidify on its own. Other times, the process of cross-linking will occur with the aid of things like exposure to UV light, physical changes like heating or cooling, or chemical changes due to the addition of certain compounds. In a way, 3D bioprinting is very similar to making a cake. First, you have to pre-bioprint. Similar to the medical scans that are used, you have to use a recipe to model your cake after. Instead of using bioink made of cells, hydrogels, and other chemicals, you make a cake batter with eggs, flour, sugar, and other ingredients. Then, you create your cake layer by layer until they all come together, just like when 3D printing. Finally, the solidification process is like putting your cake in the oven and letting it harden to achieve the desired product. As the technology of 3D bioprinting continues to be developed, more and more advances are made. Currently, we aren't quite able to produce entire organs due to the complexities of the tissue, but there have still been uses of 3D bioprinting. For example, a tracheal splint was created for an infant with TBM, and bladders have been constructed for those with end-stage bladder disease. In fact, a rabbit-sized heart was even created in 2019. Other applications of bioprinting can be creating artificial organs, bone tissue regeneration, aiding in cosmetic surgery, and producing living tissue that can be used for pharmaceutical testing without the risk of harming a test subject. In fact, 3D bioprinting is being used to create drug delivery systems that respond to stimuli such as pH, temperature, and light. Thank you for watching.
So this is just a very easy video where we would talk about the process of
3D
bioprinting.
bioprinting by Abir.
And one second, yeah. So interesting fact is that they mentioned about baking a cake and the processes that are being done. Addition point to note, 3D bioprinters are open systems that can even allow food production, food printing, and many other applications. I will be talking about each of the applications one by one. Until then, I'll just give you some examples, say 3D printing a ear, ceramic-based materials for bone development, cancer metastasis studies in blood vessels, the use of organoids for drug testing, creation of heart models, that is miniature heart models, and later on progressing towards larger size heart models. Healing injuries, grooves and grafts, implants, such examples. And then you have maybe printed skin. So the types of bioprinters, which is already mentioned in the video, was inkjet, laser-assisted extrusion, stereolithography. So inkjet is basically taking an ink material. There's piezoelectrical effect, which eventually leads to the cause of droplets. And these droplets, when they're printed, they combinatively form a structure. But here, inkjet printing is usually used when it comes to printing with materials of low viscosity. On the other hand, you have laser assisted. So laser assisted is basically pulses of laser would be going onto a layer, and this eventually forms droplets through a bed of bioink, and this again comes out in a droplet. Then you have extrusion, which is the most widely used, and it also takes a concept from FDM printing or a general 3D printing, where mechanical force is applied. It can be general syringe mechanical force, or it can be through pneumatic pressure as well. And this pressure applied eventually leads to extrusion of the bioink material through a nozzle, again nozzle of various dimension to give a structure that you want. Then you have stereolithography, also called SLA, where a light source passes and a light once it goes through a projector, it goes into a bed of bioink material and it prints in a layer by layer fashion when the light interacts with the material, that is by photopolymerization. So these are the general basic ideas behind the bioprinters, and the types of bioprinters are extrusion printing that I mentioned of. So we have a system which accommodates syringe printing, EMD printing, EMD basically inkjet printing, as well as extrusion printing, and you can also add many other additional points to it. This is supposed to be pressure assisted, filament dependent. You can easily create multiple models and also use multiple materials to do the job. Then you have digital light processing, which is a sister technique to SLA, and it is light assisted, filament independent. You can create only limited set of structures, limited materials that can be printed together, but they will give a very high resolution watertight structure. So this is one of the extrusion based systems. then you have the DLP based system. So steps that are generally involved in bioprinting, one is attaining the data or attaining the structure you want to create in terms of customization, in terms of research. That is, in some cases, it can be anatomical data which is already available. You have MRI scans and CT scans, or you have 3D scans which can be used, and this is the imaging part of it. You have 3D design. So whatever slices or whatever images you find in terms of customization, they need to be stacked up into a 3D manner, giving you a specific 3D design. And this design later comes down to using the machine, using a specific type of material. So design, material selection, cell selection, G-code formation are quite kind of dependent on each other. So you have materials which will give you good amount of mechanical strength, and on the other hand, you also have materials which are meant for very high biocompatibility but less mechanical strength, and you have many types of materials that way. So you have generally used gelatin or alginate or gelma. You have PCL material, you have PLA materials. You have different types of materials that are meant for each application depending on the shore value, that is the strength, the total mechanical strength of a material. And this is cell selection. This is where you take care of which application you are working towards, and you also have the right form, say maybe differentiated cells or induced pluripotent stem cells or non-differentiated cells. You can make that decision depending upon existing data or what you want to research upon. This is called a biodevelopment. So this takes up the major chunk of any research work, that is material selection as well as cell selection. And they ideally use the concept of material sciences, tissue engineering, cell biology, and especially life sciences also comes into one of the sectors. So the technique you're using will also be interdependent as such on what type of material, what type of cell, say extrusion-based printing or DLP-based printing, and from there you go down to the post-processing. Post-processing can be UV curing or chemical curing or temperature-based curing. End of the day, the material that you're printing will be a slightly semi-solid or gelish or a gel form of structure. You need that to be more stable, more mechanically confluent, as well as has the capability of allowing the growth of cells within it. That's why UV curing plays a very important role in certain print jobs. Then you have incubation, depending on if you're printing with cells or without cells, and you would have a span of say day 0, day 7, day 14, and so on and so forth as to how many days of incubation would be required for the cells to grow, give the required mechanical properties, assess the cell viability, and also see eventually whether it's giving me the final result that I want. So you have in vitro tests and applications. This is where the final concept comes in, whether you want to use it in as a graft, as a treatment procedure, or as just a testing platform. So applications of 3D bioprinting, they are soft robotics, biomaterial development, organ on a chip, tissue model, drug discovery, 3D cell culture, complex models, and drug delivery. So I'll go one by one, giving you small examples. This can be quick because we will have more detailed presentations coming up on each of the areas or selective set of areas. So stay tuned to that. We have soft robotics, which is the first one, which also kind of brings down the idea of biohybrid robotics. So what is soft robotics? Let me tell you that. Soft robotics is basically the idea of replacing a material which is very rigid, which is less flexible. For example, a metallic robot, right? Robots are quite metallic in general. The inner structures, the gears, the fixtures, all of that is completely specific to one specific direction of movement. And on the other hand, you want certain materials that can be more adaptive in terms of them, more complacent, more compliant in terms of the structure you want. For example, um, I want a muscle action to be replicated, or I want to use a device that can easily glide through my blood vessels without damaging the arteries or veins, and it being smart enough to reach the destination that you want without any additional issues. So having smart materials, creating smart materials is what soft robotics is. And then also comes a section where you can combine material science, biomechanics, sensing, and cells together to create more biomimetic devices or biomimicking structures. So I'll give you a few examples, vague, light examples, which can help you understand eventually what is it that they're trying to do. So this is where they've used a sugar-based material and try to print that structure. Upon printing that structure, they want to see whether it can easily stay in a solution medium or solvent medium. Will it expand efficiently, effectively? Say example, when it is kept in a specific medium, in a specific temperature, what is the kind of changes that happens to the material over time. On the other hand, we also have magnetic materials. So you have magnetic materials which can be easily printed, or something that can give a good assistive device, a good wearable device that we can use as a non-invasive or an invasive structure. Then comes biomaterials development. So this is honestly my favorite example because this uses multiple solutions of 3D printing themselves. So one being using CT and MRI. In this case, they have taken CT, they've taken the patient's defect, they have tried to understand what segment of it needs to be reconstructed. That is, if a patient has a radius ulna fracture, if I want to put a graft-like material, how can I do this? So first question is also, why am I even doing this when there are standard methods available? So the kind of materials that you would use, say TCP or hyaluronic acid or any type of bone structure, PCL, any of these materials that you use as grafts, they are in standard form in terms of beads, in terms of powder. And when I need to use it for a patient, I have to do some sort of calculations and also a process of creating that mold or creating that design that I want. So this is a tedious and a time consuming process, and that is why 3D bioprinting can help in creating effectively the CAD design model, the customizable model that you want, again with the help of certain softwares. So you have softwares like Mimics, etc., which help segment a specific structure, using that structure and creating a design using a slurry-like form of the material you want to print. Uh, Once that is done, there's also testing required. That is, how well can I do the surgery of this patient? So you have pre-surgical modeling as well. Pre-surgical modeling will require using the concept of 3D printing into it, where I will use a plastic form of the radius ulna or maybe a simulated human structure, where I will try to see how much time will it take me to stitch it up, stitch the person up, add the graft. So if I have planned my surgery well when it comes to using the actual 3D printed form, that is a 3D bioprinted form, I can easily save on time and also cause less host defects. That is, I don't have to cut up on multiple places. I will just cut up on one side, which is more efficient, more effective, and then use that in effectively installing the graft. So here they have used ideas of mechanical printing, that is how you would use infills, whether I would use um, a 50% infill, a 25%, a 75%. So the idea of infills here is basically how efficiently I can fill a solid structure to form the solid, right? Um, Either it would be something with a higher amount of gap. That is, if I do that, then I will save on material. If I want a structure which is more structurally or mechanically capable, then I might even end up using 100% of my infill to avoid that any sort of mechanical tensions are avoided. But sometimes you also have structures that can work effectively in 75 percentage, because here you won't use a lot of material, but you will also create a structure with the right amount of functionality required for it. Then you have 3D cell culture. So what generally used to happen is we would culture cells in tea flasks, in petri plates, or any type of flat structures where the cells will effectively bind to it and grow over there. Now, cells are generally spherical in nature and they are 3D in structure. So when you have the cells being placed on a flatline structure, there could be a negative effect of the substrate over the cell. Yes, there is effective growth, but the efficiency of the growth of the cells might not be the best because you have only two-dimensional interconnectivity between cells. We are looking at three-dimensional interconnectivity because end of the day, we are 3D form, 3D structures. The cells within us, how we see, how we perceive, everything is in a 3D format. So an alternate to that is creating 3D models, that is more physiologically relevant, environmentally relevant models to carry out study more better and also creating easier testing platform. So these are images of the structures being created in the 3D as well as the 2D. So you'll see there's better interconnectivity, there's better physical, environmental, physical structures, and they become more helpful in terms of understanding the mechanisms or the cell biology of the cells or the cell type we are trying to study. Then is tissue engineering. So tissue engineering becomes a very, very wide area, a wide concept, because here you can go down from creating a regenerative model to creating an aiding model, creating a basic implantable device with cells or without cells, or anything of that sort. So using a 3D printer, a 3D bioprinter to be specific, can start from maybe a basic design and then go up to complex design. So it has the capability of everything that you want. Here we would use materials like Laminate 521 and create a very basic hard model just for us to understand what could be the effect of printing with cells using a 3D bioprinter. So we would use this mixture of bioink along with the cell type and then putting it into the machine or the 3D printer into a cartridge, applying a specific type of pressure and then printing the structure you want. So the structure that you want is printed at effective pressure, effective parameters, speed of printing, all that is being kept, and you incubate the structure at a cell medium for a span of 3 weeks or so. So what you see here is an image of the spherical structure, the spherical viable cells that you see on the given tissue model that you want. And you would see here that they had a calcium influx, just to show us that it has a capability of replicating something as a basic muscle cell, a heart cell that could be used eventually as maybe a patch-like material, that is in terms of treatment of any cases of myocardial infarction or creating a specific layer, trying to create a valve, a vessel, maybe a ventricular structure. So there are many, many ways how you can use 3D printing or tissue engineering concepts into creating specific model types. Then there's complex design. So complex design is basically trying to replicate something very miniaturized, very complex within the body through an external structure. So here they have taken heart model, and this is a patient-derived model. The idea of this complex design or this case is where they're trying to understand the effects of using any type of imaging device, imaging method, use of bioprinting, as well as the effect of congenital diseases, or how are congenital diseases formed, or how could one reverse the effect of specific disease, right? So here they're taking the concept of embryonic development, fetal heart development, whether it comes to creation of the tube or creation of the ventricles. So the material used is Gelma, which is a proprietary material from the company Cellink, which works with the Luminex Plus machine. Along with that, it has the materials that can incorporate effectively in terms of printing the structures. So the structure which is printed is, say, the CAD models that you see, and to them we are trying to seed cells and see what are the environmental factors over it. Try to replicate perfusion. see what mechanical impact would fluid flow within the body lead to in terms of the cells collection or maybe development of any structure, and doing the analysis. So this type of method can easily work out with many such issues. So say atherosclerosis, you want to replicate a complex vessel model, you want to do a pre-surgical model using a soft material as a base. So there are many such sectors that we can get into. Then you have organ on a chip. So organ on a chip is basically creating a one chip model which can easily be a testing platform in the pharmaceutical workspace so that you can replicate the physiological environments required for a cell type, basically a liver, a lung, maybe brain model, maybe neurons, liver model. So whatever examples I would suggest, the respiratory tract. So you want to see what can affect the specific structures and also how drugs can help in improvement of these structures. So you have a testing platform. Here it incorporates the use of microfluidics because microfluidics is what essentially allows the reaching of specific material or specific cultural media to the cell type that you want, allowing better perfusion, allowing better oxygen carbon dioxide movements, creating a better reservoir. So all of this also plays an important role. Then we have drug discovery or high throughput screening. So this is a very interesting factor because high throughput screening or drug discovery becomes an essential part in the pharmaceutical workspace, taking the metabolism, checking the pharmacokinetics, the toxicology, impact of the drug on a cell type, which is maybe healthy or unhealthy cell type, plays an important role. So this case is where they're trying to do drug-induced liver injury tests, where you're trying to do a predictive toxicology model. So if you say, for example, you have a specific model, that is say, I want liver model to be created, they try to print the liver model. They will check the collagen deposition, the ECM production, the ATP levels, the albumin levels in a span of 4 weeks. And now they will try to create a testing platform, that is this liver, to easily allow them to check what could be the prediction. That is, say, for example, I have a drug of family A, and in that family A, I have two variants, that is family 1, variant 1 and variant 2. Now I want to be able to see what could be the effect of variant 1 and variant 2 in their own specific ways, or you can give a prediction, a predictive correlation between different types of drugs and how they impact the cells, whether it is healthy cells, whether it is unhealthy cells. So this is where precision medicine, you have disease modeling, you have cancer model studies, all of that come in that area. So you have a lot of companies, a lot of people who are coming into the healthcare or the life science space, working with studying the carcinomas, that is using the concept of 3D printing, that is by creating an oncology model. So the kind of leap 3D bioprinting has been taken in the pharmaceutical space is been from creating very simple 3D models to creating complex multicellular models, just doing toxicity screening or checking the impact of one drug on multiple areas, or doing precision medicine, that is a patient specific. Initially, you have the allogenic pool of cells that could be used from various people that you do the testing for, but there'll come a point where you would want autologous cell types, that is patient specific cell types, to do the treatment of the person. Then comes drug delivery. So drug delivery is a concept of effective, novel, smart drug transport to the body. Our idea of drug delivery is targeted and sustainable drug delivery, and that's how using biomaterials, using 3D printing concepts can help us. So this is a very basic example where they've tried to create a biopierce for a ear stud, a piercing stud, where they wanted to avoid the use of medications, oral medications or ointments, and instead putting a wearable device, a wearable structure to your piercing point, and it would allow sustainable release of drug at that point, avoiding any sort of infection. Now, this is very basic. You also have customized drugs. You want to create an implantable drug device. You want to create some sort of pouch, some sort of devices where you can easily fit in the body while being biocompatible enough is also another point. And So yeah, there are studies being done in terms of the zone of inhibition, the amount of effective drug release, what is the comparison between a standard model, for example, a placebo or a general, and control groups are checked, and all of that are done in general. So that's how drug delivery would work. Now the solutions that selling provides are quite varied, one being using the basic model structures, that is the pneumatic based printers. You have complex model structures, the BioX, BioX6. Then we have the BioMDX and the BioMDX Plus, which is an industrial device. So you have each type depending on application. Now they all cater to all the applications. It's just the scale of production or the scale at which you want to use it. Also the type of precision. So the DLP systems or the light-based systems are Luminix Plus and the Quantum X Bio, which is a new device that we have put out. This is meant for microfabrications in 50 microns or less than 50 microns, 10 microns level of precision. So if you want more details on this, you can easily visit our website, Altum Technologies, where you have technical specifications of each of the devices that is based on your interest on your project. Then we also have BioVia, which is handled by my colleague who takes care of, in silico analysis. So we have BioVia Discovery Studio, which is capable of protein engineering, antibody modeling, simulations, predictive ADM tests, X-ray analysis. It goes into the realm of bioinformatics, cheminformatics, say virtual screening, genomics, everything in the bioinformatics space that can come before in vitro studies. So in silico studies would be the main crux of it. So thank you so much. I think I have covered all the basics within a given frame of time, although there are many more things. So I'm hoping that in case I see less people here, at least in the next 4 sessions, I would see more people because we have some experts from the industry who would give us your insights, who would give you, answer your questions depending on what field you're from, and also have interactions with them. So thank you, and I hope to see your, responses on Facebook, YouTube, LinkedIn, as well as Twitter and any other module. Yeah. So I'm just looking for any questions if there are. So in case no one's able to type down the questions, or if I'm quite clear, then maybe you can write to us, or you can, 1 second, I'll just see if I've missed on any questions. Yeah, so I don't see too many questions for today. Any unanswered questions or any questions you have not put down, we can take them for you tomorrow. You can send us an email on Altum Technologies, and I hope you like the session, and I hope to see you in the further sets of presentations that we have. Thank you so much. So I will end this for now. I hope I see more people in the next few ones. Day 3, day 5, we have Dr. Shreya Malhotra, as well as doctors from IISc. So thank you so much.